Welcome to Pharmacology of the Synapse. This week we are going to learn about neurotransmitter release machinery and how these molecules can be disrupted by toxins. In the unifying figure, our topic this week will focus on synaptic neurotransmitter release and metabolism. We will examine how natural toxins generated by plants and animals affect these functions. The synapse we will be talking about as the prime example for vesicle release is the neuromuscular junction. To activate muscles, a motor neuron contacts a muscle fiber, and this contact is called the neuromuscular junction. The end of the neuron splits into many presynaptic terminals. Within the presynaptic terminal, vesicles containing acetylcholine line up to be released. Depolarization of the terminal causes a large amount of vesicles to fuse and release neurotransmitter. This causes muscular contraction. The activity of acetylcholine is terminated by the metabolism of the neurotransmitter by acetylcholinesterase. This figure goes over the key points within an acetylcholine synapse. Acetylcholine synthesis takes place in the nerve. It is then packaged into vesicles by a vesicular acetylcholine transporter. After release, acetylcholine is immediately degraded by acetylcholinesterase, and this action terminates acetylcholine signaling. Acetylcholine is broken down into choline, which is transported by the choline transporter back into the nerve. Additional players in the synapse include presynaptic receptors and postsynaptic receptors. This figure illustrates a typical synaptic connection between nerves. If this were a neuromuscular junction, all the components would be the same, except that the postsynaptic muscle side would have many nicotinic receptors on the surface to depolarize the muscle. Next, we're going to take a look at the machinery that releases the neurotransmitter into the synaptic cleft. This is a close-up look at vesicle fusion, and the two major components required for vesicle fusion and exocytosis are SNARE and SM proteins. Thus, this is called the SNARE-SM protein cycle. The diagram on top depicts SNARE and SM proteins prior to fusion, when they are localized to the membranes either as natively folded proteins, for example, the R-SNARE synaptobrevin protein in red, or as proteins folded in complexes, for example, binding of the MUNC18 protein to syntaxin 1 in teal. During priming, SNARE proteins partially zipper up into trans complexes, and the SM protein associates with the trans complexes by binding to the syntaxin amino terminus. Full SNARE complex assembly then pulls the membranes apart, opening the fusion pore, which expands such that vesicle membrane collapses into the target membrane, and the trans SNARE complexes are converted into cis SNARE complexes, meaning that all of the components are now on the same membrane instead of on opposite membranes. Afterwards, cis SNARE complexes are dissociated by the ATPase called NSF, acting in conjunction with its adapters, alpha, beta, gamma snaps, and the vesicle recycles to start another round of release. Here is another close-up view of the synaptic machinery. You can see the components come together in trans complexes in the middle section, where the complexes are formed from proteins on different membranes. Full association of the complexes pulls the vesicle membrane and plasma membrane apart to form a pore where the neurotransmitters can be released and the vesicle membrane fuses to the plasma membrane. In the next section, we'll see how toxins disrupt the vesicle release machinery.